Hi, everyone. I'm Dr. Kelly O'Donnell. I am the Director of Science Forward at Macaulay, and with me is Dr. Lisa Brundage, the Director of Academic Affairs. And we want to tell you about how we took our BioBlitz online in 2020. We are going to use our time this morning to talk about how we took this essential experiential learning experience, experiential learning event that's an essential part of our science curriculum, and adjusted it to the online world of 2020. We want to go over the goals of what the goals of the BioBlitz, what we had to do to make that transition and still keep our students engaged, how the BioBlitz works in the classroom, and the lessons that we're taking forward for future BioBlitzes. So the BioBlitz is an essential component of our Science Forward course at Macaulay. Science Forward is a skills-focused science curriculum. It's also an open educational resource that you can find at the link down there. All of our students have to take it regardless of what their major is, and we make the course interdisciplinary so that students see that there is a common set of skills that all scientists have when they do science. We want those students to practice those skills in an active classroom. And because this is a science class that has no lab attached to it, we need to have that sort of science experience for the students to be engaged in the process of science and learn how science works by doing it. And that is what the BioBlitz is for. So we start every year in the fall. All of our sophomores take this in the fall, and we start the semester with a BioBlitz. Now, a BioBlitz in general is a species diversity survey that takes place in a short time period in a restricted area. So in our case, these are often public, like citizen or community science type events where lots of folks who are not necessarily professional scientists come and do this species diversity survey together. So in our case, our BioBlitz is a 24-hour survey of a park or other open space in New York City. And all 500 of our Macaulay sophomores are required to participate in it. So in a typical year, not a pandemic year, so not 2020, our students sign up for 3-hour shifts, and they're put on, so they're not there for the whole 24 hours. We're there for the whole 24 hours. And they sign up for 3-hour shifts. They're put on teams with taxonomic experts that we call taxon leaders. And these are naturalists or professional scientists or even hobbyists that have a really good knowledge of our local flora and fauna. And those folks lead our students around and demonstrate good observation techniques. And so our students are set on all these teams. There's a bat team, a bird team, plant team, fish team, lots of different teams over the 24 hours. They collect data on paper, and they collect data using the Inaturalist app, which I will talk about in a minute. And those data are compiled, and then they're sent to our faculty. We have about 30 faculty who teach Science Forward across the 8 campuses that have Macaulay students on them. And the students use those data to do a semester-long research project that they then present at a conference at the end of the semester called the STEAM Festival. So students spend time in class creating their own research project based on these data, and that gets them engaged in the entire process of science, from developing their own questions, picking the appropriate analyses, and communicating clear results. So the BioBlitz event itself achieves 3 goals for us. It generates data that the students can use in class. It has students learning about how scientists do their work in the field, so students are engaged with scientists. And it connects our students across the 8 campuses because they're all there together. We shuffle them on the teams, and they're all out collecting the data together. Now, for 2020, because of the pandemic, our Macaulay classes, we couldn't have any in-person gatherings, even if they were outside. And there wasn't, even though this is outside, there simply wasn't a way to hold it safely and get our students to and from it. And so we had to make an adjustment. We had to redesign the BioBlitz to be completely remote while keeping these 3 essential goals. And so that's what we're going to talk about now. So the most important goal for the curriculum itself is getting that data set, that student generated data set that can be used in class. And because that is how we get students to, take ownership of a science project and actually do the science themselves. And luckily, we were already using a tool that would facilitate the transition to a remote BioBlitz, because we were using Inaturalist. And what Inaturalist is basically, it's a free app. It's connected to a web database where you can take photos of nature, of species in nature, and get the community to help you identify them. So you don't have to know what you're looking at. You can just take a picture of something, post it as an observation, and then the iNaturalist community will come in and help you give it an ID. So as the user, you're uploading geotagged and time-stamped nature observations. And so these can be associated with a place because they're geotagged, and iNaturalist has this function called projects that can automatically aggregate data based on a place. So usually at a typical in-person BioBlitz, we would have this project set up with the boundaries of the place we were surveying. As the community agrees on these observations, the species that are in your observations, they become what iNaturalist calls research grade, which get, and those research grade observations get fed into the Global Biodiversity Information Facility, which is a place where scientists can actually go and pull down data. And to answer all kinds of questions about ecology or distribution and abundance of species, and do science. And so in this way, using iNaturalist connects our students to both, the iNaturalist community of users itself, but also this sort of global community of scientists. And so for the remote BioBlitz in 2020, this is what the project looks like here. You can see a summary of the results here, the observations that the students made. It also has leaderboards for most observations, most species, and which species was the most observed. Our students who registered for the BioBlitz had to make an account on iNaturalist, so they were all associated with this project, and we tasked them with making 2 observations in the month of September. Quite a few of them made more than 2 observations. As you can see, that's how we ended up with 2,399 observations. And one of the benefits of using, so here's some of the results you can see here. One of the benefits of using iNaturalist and using these iNaturalist projects is the automatic data aggregation. And since our students, and you do that by setting up these project requirements. Now, since our students were distributed and not, they weren't all in the city, they weren't all in one particular place like they would normally be at the BioBlitz, what we did with the project is that it wasn't a place-based project anymore, it was a user-based project. And so all of our students became users here. Became basically project rules. So all 500 of our students' usernames were here. And what this thing is doing is basically any observation made by any of these users, no matter where they were automatically pulled into this project. And so this is what generated the student data set. And this was, another way to connect our students together. They were all in this project together. You can see they found, there we go. In terms of the data that we collected, we had over 2,000 observations of 736 species across 5 countries. And I mean, most of them were in the US, but we did have 5 countries spread of observations. 96% of our sophomores registered for the BioBlitz. And then of those that registered, 93% uploaded observations. And so this is typical, like similar participation to what we would have at a typical in-person BioBlitz. Usually we get most of our students come and participate. We have very good attendance here. And so we had very good participation comparatively at this remote BioBlitz.
Okay, so I'm going to talk a little bit about how we got good participation and how we engaged our students. When we do an in-person BioBlitz, it's a very logistically complex and very visible event for our students. You probably saw from some of the pictures from past BioBlitz that Kelly shared, they have t-shirts, they come in, there's a lot of kind of messaging and enthusiasm and like hype that we build around it. And because we have now been doing this for a number of years, our students know that it's part of the Macaulay experience that they participate in BioBlitz when they enroll. And so it's something that they look forward to as the beginning of their sophomore year. So I wanted to just point out, I dropped a few links in the chat to our BioBlitz site that we maintain you can pull down all of the data sets from our BioBlitzes. We maintain all of our data from the paper data sheets as well as the iNaturalist observations, and they're all openly available. And I also put in links to our iNaturalist project pages, one to all of our BioBlitzes and one to this year's remote one. So because this is a really big event and we weren't sure when we were setting our budget for the 2020-2021 school year, whether we were going to be able to do it in person or manually, we were very lucky in person or online. I don't know why I said that. We were able to make some care packages for our students and send them out. So that was sort of the first round of making sure that they had something that was tactile and that they could really like see like, oh, this is like a big deal to the school. This is something that they really want us to do. They got in that care package a little folding sun hat, a ruler that is really useful for scale in those pictures that are going onto iNaturalist, a little hand lens that you can see over there on the left, so that if they were looking at tiny things, you can hold a phone right up to the lens there and you can get a picture of it, or you can just use it to look yourself. A nice sticker. And they also got, there in the back is a postcard that on the other side of it has instructions for what to do. So in case they were missing our electronic communication, that they would also have that they could look at. And we also had these nice little packages that our colleague who couldn't be here with us today, Janet Fu, personally packed and mailed all of these packages to 500 students, including a really cute little sticker on the front so that when it arrived, they knew that it was coming from Macaulay, that it was going to be something kind of fun and a little bit different. If we didn't have the same kind of budget to do something like this, even the postcard by itself going out could make a big difference in terms of reaching students and catching their attention a little more than the voluminous number of emails that they always get, especially this year with everything being remote. The other thing that, well, not the other thing, but one of the other things that we did, Kelly, you can go to the next slide. We do a lot of communication with the students around it. So in a regular BioBlitz year, we had already this established email, just bioblitz@macaulay.cuny.edu. Somebody checks that all the time. In a normal year, we have questions like, I signed up for this shift. I want to go to this shift. Can you change this for me? Can you make sure that I don't have to see a spider? What should I do if I am worried about, you know, I don't know, we've done BioBlitzes at places like Fresh Kills and Gowanus, and sometimes they have questions about the environment there. And they know that they can email this and that somebody is going to get back to them pretty much right away with information about BioBlitz, whether it's taking care of their registration, answering a question, asking, you know, what they're supposed to do once they arrive there, anything like that. So this year we still maintain that. We had to register them for all the webinars that Kelly is going to talk about soon. And so they have, you know, a really open, clear line of communication to us. They don't have to think like, should I email Lisa or Kelly or Janet or my professor or somebody else to figure out how to get information about this event. So that's been really successful for us. We also, of course, used our electronic means of communication through the college's official channels. So we featured it on Instagram, especially we encouraged students to take science selfies when they were out doing their observations. So here we have one of our students who's wearing his BioBlitz hat and is out. Actually, he had experience working with fish. So he was looking in a local pond for aquatic life. But we made posts about that. We had a hashtag for it. We aggregated all of those posts. We also have a newsletter that goes out to our students every Monday, Macaulay Monday. And so we were highlighting in Macaulay Monday interesting, they're all interesting, but unusual observations, people who were really exceeding the 2 observation requirement and doing a whole bunch of posting or identifying of other people's observations on iNaturalist, and making sure that we were promoting like enthusiasm and joyfulness and a little bit of playfulness around doing this event with them.
And so, as Lisa mentioned, these webinars, replicating the interaction with scientists was a little bit trickier to figure out. But ultimately, we decided we'd have these 7 webinars that featuring panels of virtual taxon leaders. So the folks who would normally be with our students and leading these teams, we had a series of webinars featuring them. Lisa made all of these delightful title cards for the webinars. And I couldn't fit all the panelists in there, but just so you get an example of who was on these. And these are, you can see photos of people at our previous BioBlitzes and some new folks as well. The fact that we were doing this remotely meant that we can bring in some taxon leaders from the past who were really great, but who had moved away from New York. So it was nice to reconnect with them. And what these webinars did was each of the panelists introduced their organism or their group of organisms of interest, how one goes about finding those organisms and making good iNaturalist observations of them. And what I mean by a good observation is one that the student can take a photo of it and they can get help from the iNaturalist community to ID it, because our students generally don't have a lot of taxonomic experience. And we want them to get that interaction. We want them to get species IDs. And, there's some tips you can do to make your observations better so that people who are just looking at the photos can help you identify them and give you like actual down to the species level identifications. And we also had these virtual taxon leaders make a little or send us video footage of themselves in the field, so we could also get the students a chance to see these people sort of in action. Normally they would see them, they'd be there in person with them, but these little videos allowed us to just show these folks in the field and how they sort of interact with their organisms of interest. Back to Lisa.
Yeah, so there are obviously a lot of parts that need to come together to make a project like this happen. And so I want to talk about what our centralized responsibilities were for this, because something else that's good to know about Macaulay is that we are a consortial program. We operate on 8 different CUNY campuses. And we also, the faculty that teach our courses are drawn from those 8 campuses and chosen by their local Macaulay office. So we need to kind of get in our professional development and community building for our faculty where we can, and BioBlitz is one of the ways that we do that. We always try to emphasize that since this is an event that we require, we take on the heft of the work so that they are well set up to use the event in their class and really make it part of the learning experience and not part of the planning out and making it happen. Our faculty are always welcome to come to the event just as participants. They don't have to do anything to make the event happen. But since this year we were going to be doing everything on iNaturalist, we wanted to make sure that our faculty were promoting that. Doing everything remotely on iNaturalist, not in a concentrated weekend altogether. We wanted to make sure that our faculty were ready to participate using it, also to promote it to their students, and then knew how to access the data. So we set up the iNaturalist project that Kelly showed and did that, as she explained, in a different way than we usually do, since it was run by, it was delimited by users and not by location. We did all of the planning and the registration for those webinars that Kelly just talked about. We sent out all those packages with many thanks to Janet. And we also just made sure that we kept those lines of communication open all of the time. The final thing that we did is the culminating event STEAM Festival that we have every December. In the fall, all Macaulay first year students are taking a class called the Arts in New York City, while our sophomores are taking Science Forward. And we always have a big event where they come and share all of their projects at the end of the semester. That means it's about 1,000 students. They usually come in, again, over the course of a weekend to the Macaulay building, where we have all of the rooms set up as poster galleries and like art galleries. So this year we did that also online, and I'll show that in just a second.
And so to get this work sort of seamlessly with the classroom experience, as Lisa mentioned, all the faculty had to do was put this in their syllabus as a required event. Like, your students have to go to it. You don't have to go to it. We welcome you, but your students have to go to it. And they have to use the experience somehow in class. Now that can be anything from, and so on the Science Forward OER, we have this list of like possible ways they can use the BioBlitz experience. You can talk about what it means to collect data systematically versus at a citizen science event. You could talk about AI stuff because iNaturalist has an AI species suggester. You can use it to make infographics. But the thing that we most strongly encourage is that the faculty use the BioBlitz data and the BioBlitz experience as the basis for a semester-long research project that they end up presenting at the STEAM Festival. And so in order to do that, we had to have some workshops over the summer about, what you can do with these data, basically. So basic ecological analyses, suggestions for activities. This is not a stats class, so it's not going to be like an in-depth, and also these data are very messy, which is a very good discussion to have with your students to build data literacy, because talking about the way these data are collected and how it might be different from like a more systematic ecological survey is actually a really good discussion to have with the students. And so what we want for the students and what we provide for the faculty, we provide a guide for the scaffolding of this activity as heading towards a research poster based on the BioBlitz. Because what we're trying to do, again, this class has no lab, but it's a science class, and we want the students to do science. We want them to have an authentic research experience, because that has been shown, there's evidence that shows that this improves critical thinking. It improves the understanding of how students understand how scientific research works and what the purpose of scientific research is. And there's sort of, you poll a bunch of faculty and you get different levels of importance for different themes that fall into what is authentic research. But most of them come up with some list like this. So, and we want our students to do this. So the students have collected the data themselves. The students come up with a question that they can ask of those data, and then they have to pull down the relevant they have to query the database and pull down the relevant Inaturalist data that would answer that question. So they're designing the analysis. Then they are, building that presentation and clearly communicating the results. So they're doing this entire process of science. They're getting this authentic research experience. And we were still able to do this with the remote situation because all of our data were on Inaturalist and openly available. They also have access to all of the past BioBlitz data because those are also all open. And we compile them in an in-person BioBlitz, the paper data sheets. So we actually have this huge data repository now from our BioBlitzes and from anything you could pull from Inaturalist because it's all open. And so you can see there's a couple of student poster examples here. The students get ownership over this work, and they've done this work themselves. They're the scientists, and that's really what we want them to get out of this whole experience.
Right, and finally, I just want to talk about a couple of things that we are going to keep moving forward, even past the pandemic. So because we because it's so resource intensive to do the BioBlitz and the STEAM Festival in person, we've always thought it would be great to have more online engagement around those events, but we really haven't had the, we haven't had the space to build the infrastructure for that, and now we have it because we did it this year. So we want to do some more engagement of students with Inaturalist outside of the BioBlitz. We always have them come into the site for the BioBlitz and kind of do, a crash course really quickly and using it, but we want to host some webinars on Inaturalist techniques so that students who are really enjoying it can kind of continue to use it in their lives and throughout a longer period of time. We also, for our students, the scientist engagement part is so important. A lot of them are STEM majors, but they are very focused in the way that they think about science in terms of lab work. And so this is for a lot of them the first time they are doing any kind of like real field work with ecologists and getting muddy and going out and really getting into the urban environment. So we want to continue to have the webinars because it was a place for some longer engagement with these scientists, and they could talk a little bit more about what they do when they're not, also leading a group and kind of showing them how to actually do things hands-on. So we want to be able to, link those things in the future. And the other thing that we want to keep on doing is having this digital archive of our STEAM projects. So here's just one that was a BioBlitz related poster, and I'm putting in the chat a link to all of our BioBlitz work that happened for our virtual STEAM festival, because it was really great there to be able to navigate through the site and see some of the through lines between the art and the science projects, and also to have a little bit of an easier way to aggregate everything that was related to BioBlitz. Rather than moving around the physical building and trying to hit all of them, now you can pull them up and take your time with them. So I think that we learned a lot this year about how we can make this successful, and also about how we can augment what we do going forward post-pandemic. And we remain as excited as ever to do our BioBlitzes.
So if anyone has any questions, we are happy to talk BioBlitz. Or integrating iNaturalist into your curriculum, because I am also all about that.
If no one else has a question, I even as a moderator, I have a question. I think this is
Yeah.
very fascinating. Um, from my perspective, we train teachers in using online systems to teach, and there's a bigger push towards active learning. Now, finding examples in the sciences has been very difficult, like specific, substantial examples. And I wanted to know, how do you feel this affects the foundation of scientific method? What I mean by that is that um, science is based on repeating an experiment, and the money is for doing an original experiment, but not having repeated an experiment. The idea of confirmation and consensus being reinforced at such a fundamental level seems very significant. Have you considered this? Is this, what could you say to that?
I mean, that's a discussion that we have in class frequently, even and kind of related to the BioBlitz too, because like, you know, we talk about what it means to collect these data and what they would be useful for. And you know, these data, these are community science collected data. They're not a systemic, like you're not running transects, you're not setting up quadrats, but it gives you information about the world, right? And it gives you information you didn't have before. And that for our students is often an important discussion to have, because we'll come out of the BioBlitz and they'll do these posters and they'll be like, well, I didn't find any significant results, or I didn't find a trend in these data. And they think that they failed. And we have to have, every year we have to have this discussion, like, no, you learned something, you learned, your data told you something, it told you there's no relationship. And that is information you didn't have before. And so talking about those negative results, which don't get published, and so you start talking about publication bias, and then you get into all of those things about what gets funded and what doesn't, and why we might have a problem if things like negative results don't get funded, if confirming experiments don't get funded, like what that leads to as sort of a broader picture of the body of scientific knowledge. And that's a really important discussion to have. And I think that this kind of activity reinforces that. Like any of these like community science projects where you have students that are contributing to these databases, like it's important to talk about what those data do, like what people do with those data. And you know, if other projects could be funded that collect similar data, would they, and why or why not? Did anyone participate in the City Nature Challenge this weekend? Built on iNaturalist, hosted by Macaulay. It's like a 4-day BioBlitz of the entire city. Yes, I was with Lisa. I know you did.
I'm just grabbing a link to the City Nature Challenge results.
Official results get announced on Monday, but New York did very well. There's no winning city because it's a pandemic. We're all working together, but there were 418 cities participating over this weekend all solely on iNaturalist, and part of our BioBlitz actually was based on some of the things that we've done for City Nature Challenge, because that is entirely remote all the time.
Yeah. And I also just want to add that I am not a scientist. My field is English. I didn't become involved in doing, scientific literacy work until we took on the Science Forward project in the BioBlitz. And I think that even for me, it's really changed the way that I interact with the city. I'm always looking for things to put on iNaturalist now and thinking about what it means and how anybody can really participate in those processes. And it's been it's been really great to bring that into my life, and I'm thankful for it.
There's also, I see right. To wrap up, but in terms of integrating iNaturalist into class, like, you know, even if you don't have devices that are connected to iNaturalist, if you have a computer access to a computer lab, you can take those photos other ways and still get them on the database. And so if you have like mobile device access issues, you can still get onto this database and use it. There's ways around that. And if anyone has any questions about that, I am happy to send me an email And
there are many ways to do this for free with very few barriers. And if you need help troubleshooting that, just contact us.
I'll put emails in the chat. Thank you all for coming.
Thank you, everyone.